Hi, my name is Dave Hoon. I'm from John Wayne Cancer Institute, Director of Molecular at John Wayne Cancer Institute in Santa Monica, California. I've been doing molecular oncology translational research on human solid tumors for the last 21 years. Our laboratory and our department is strongly oriented towards translational research related to molecular biomarkers in solid tumors as they relate into phase 3 and phase 2 US and international clinical trials. So what I will do is give you an overview of some of our key works. I will use melanoma as a model system, and then I will add some other cancer work that we have at the end of the talk. You can send your questions during my talk. So this is an overall structure, more like a functional structure. The genome in terms of the chromosome all the way down to the DNA. What we're going to focus on today is particularly on the methylation aspect, or in other words, epigenetics, which has a very significant influence on regulating gene transcription. More important in cancer biology, especially in the last 5 years, where this field has significantly exploded. So this just shows you epigenomic generalized the chromosome. You have a nucleosome. You can have different ways of inactivating genes, and it is as we become understanding the structural relation and the related functional, we see how genes turning on and off is very complex, but it gives us alternative understanding of how genes are actually turned on and off rather than the traditional mutations or the deletions that we have heavily focused on in the past. Epigenomics is basically the study of heritable changes in phenotype or gene expression caused by different events. There are many factors that can modify epigenetic events. That's in itself a large topic, but events that could influence carcinogenesis or even tumor progression are diet, environment, age of the individual person, growth and cytokine factors, regulatory methyltransferases, which I'll discuss later, hormones, I'll talk about at the very end, just plain DNA maintenance replication events, which occur all the time in every cell division, but it is also very critical that it is changes events, especially in the cancer situation where it can be very abnormal, and then we relate to many dysfunctional gene activity. So this is a model of metastasis. As many of you probably know, you have primary tumor growth. It can spread through blood vessels. You can have circulating tumor cells, micromets and lymph nodes, and then it can go to different organs. And so you have many aspects of events happening, and it's very complex in itself. And what I'll try to show you is how epigenetics particularly can influence this overall. So here's an overall factors for metastasis, and you can have the genome, as we well know, which has mutations, loss, copy number, and variances. And we have epigenome, which consists of microRNA, methylation, and now we know histones also play a significant role. And these all relate to the regulation of transcriptome, which is mRNA. So you can see that there are two events, major events that are happening. In the past, we have strongly focused on the genome of the cell, and now we know the epigenome probably plays equal or more influence than, especially in cancer, than ever before. And so this is what our focus is. And the transcriptome and proteome is essential that we know there's significant changes in tumor cells versus normal cells. So the gene regulation epigenetic is often focused on the CpG island, methylation status, and the gene promoter region. Doesn't mean that's the only region. It can be outside the promoter region, such as enhancers, but it is the major topic I focus on. The other is microRNA, which is an epigenetic factor, can also regulate gene transcription. And the other is chromatone, histone protein, methylation, acetylation. I won't be talking about that today. So this is just a schematic where you got exon, promoter region, and these lollipops are basically the CpG islands or sites. And if they're open, that means they are unmethylated, where red means they're methylated. So in a non-methylated situation, you would have activation of the gene, particularly open reading frame, and you get transcription, mRNA. The right-hand side, after methylation, you get hypermethylated, where you can see the lollipops become methylated, the red, and it turns off the gene, and basically you get no mRNA transcription. And what causes this is, we know one of the major enzymes is the demethylase transferase 3A and B through SAM activation of the methyl group. And this is what the methyl group looks like. The cytosine, which is your basic pair, and you get methylated, and you can see the 5-methylcytosine. So this is in a simplistic way what happens when you get 5-methylcytosine activity. Now I'll go into melanoma and to describe some of the studies we have done, but I will just give you an overall. Melanoma, as you probably know, is primary skin cancer, the most lethal type, and most of the melanomas that are found are primary melanoma. But then it predominantly spreads to the lymph node, and you have lymph node metastasis, and that's where you get stage 3, A, B, and C. It depends on the level of involvement of the node. Stage 3A and B are often sentinel lymph nodes, which we originally developed at John Wayne Cancer Institute almost 2 decades ago, and changed the practice of early diagnosis of regional metastasis. Then you get distant mets in stage 4, which is the most lethal type of the disease. And once you have distant mets, it is very difficult to manage, and is very lethal with a survival rate. The most important thing I want to emphasize in melanoma, lesion could be less than a couple of millimeters, and it could be deadly. Lesions that are like 3 to 4 millimeters, patients have a very, very poor survival rate in less than 5 years. So it's not like other carcinomas where a 1 centimeter tumor may not be that lethal. Or melanoma is an extremely aggressive tumor. To spread any site in the body, there is no preference. And go to the lung, liver, brain, skin, any organ. It's not like other organs where there's often preferential site or that is related to metastasis due to drainage patterns, like colon to the liver. So I want to describe in a study that we published several years ago. These were melanoma tumor-related genes, RAR beta-2, RASF1A, which we described in the early 2000s, SOX, GATA4, WIF1, Wnt inhibitory factor, TFR on 2, which is a regulator of plasmid. And we assess the methylation pattern through the promoter region, which we identify the key CpG island methylated sites. And this just shows you a schematic. It was done by MassARRAY. Then we look for methylated CpG island sites. As you can see, you have yellow, orange, and red circles in the diagrams under each gene. Those are the CpG island sites, and those that are highly methylated, they are yellow, and those that are not methylated at all are red. You can see next to it on the right-hand side, the mRNA expression level correlates. There's RSF1A, you can see ME7 is highly unmethylated, and you can see mRNA, where the others are very methylated and there is hardly any transcription. SOX1, you have our controls are listed, UUC and UMC, but very similar pattern, where SOX1, they're all highly methylated and there's no transcript. So this gives you a sort out. But most critical, which has been an issue in the past, is to identify actual CpG island sites that really regulate mRNA transcripts. And in order to do that, you need to do these kind of studies and profile and assess to determine what is the key regulatory CpG island site. So we did this to profile all our genes. And then we also look at what you call non-coding genes, previously known as junk DNA, where you have sequences like MINT, which are multiple cancer related, and especially MINT 17 and 31, we identified in melanoma, and these can be methylated or non-methylated, and they can act as also an indicator of progression. And this is a correlation we did with methylation status of the MINT loci versus the individual genes, and you can see there's some good correlation. So we use these as kind of a general, broad indicator of what the methylation status of all the genes are that we were assessing. And what our study did show, it was what we call SIMP, which is a methylated phenotype in melanoma. Previously, SIMP was originally defined in gastric and colon cancer, where you get high methylation as a progression factor, and SIMP phenomena. And this also was found in melanoma. However, one does still have to take this with caution, because you also get genes that also become hypomethylated. In other words, in the opposite direction, which also, but in general, the methylated tumors become SIMP, and they become more progressively methylated as it progresses. So what we did is then we looked at an array of stage 3 tissues, and as I previously indicated, DMT3A and B regulate methyltransferase activity, and this just shows you in a study that the stage 3 and 4 have much higher DMT3A and B compared to stage 1 and 2. In other words, we saw the methyltransferase significantly increases during tumor progression. And this just, then we verified in our tissue microarray of stage 3 lymph node metastases that were well clinically annotated, where they had greater than 5 years good survival, no reoccurrence, versus poor, those patients who died within less than 2 years. And you can see some of the staining and the differentials from A, B, and C. So one of the factors that we found was that a microRNA was actually controlling the methyltransferase. So here we have two epigenetic events that are working together that are controlling, and the microRNA 29 family has three members, A, B, and C. Two are on chromosome 7q, and the other is on 1q. And we found that they were very targeted to the DMT3A 3' UTR region. And what we did is, I showed you before, this is the pathway, and microRNA 29.3 was actually downregulating DMT3, and as a result, methylation of the promoter region was shut down. So here you show where the host has a way, the cells to regulate methylation is by DMT3 transferases, but it also has microRNA as a checkpoint. And so we assessed this in melanoma to see what was this relation during tumor progression. And here's an example where we saw microRNA 29C, where we did analysis at different stages, and you can see in stage 4 overall it was decreased compared to the earlier stages, which means when microRNA 29C is decreased, methylation is increased. And so when we did on the TMA, we did a staining and assessment of microRNA 29C, high-low, and on the left figure, you can see when microRNA 29C is low, you have a much poorer prognosis versus high. Understanding that low microRNA 29C has more DMT3, as you can see on the far right, low DMT3B is a much better prognosis versus high. And similarly, where high microRNA 29C versus low is much lower. So what this shows you, how these two epigenetic factors are actually regulating methylation, but they have a significant influence on disease-free survival and overall survival. So this is actually one of the first times any group has ever really shown how these two factors regulate methylation, but have a very significant relation to tumor progression in a fairly long-term 5-year follow-up. So the next thing I want to talk about is the progression from identifying these methylated markers. So we have a long history in our department is looking at circulating DNA, cell-free DNA. This is actually found in serum or plasma. And about over 10 years ago, we were able to look at specific methylated DNA in the serum that survives. How it survives for a prolonged period of time, we don't know. And how tumor-related genes actually have a better survival rate than in normal individuals' samples is still unknown. But we suspect it's the way that tumor cells go under apoptosis or necrosis and how they shed the DNA uncut, in other words, not gone through the apoptotic process to degrade. Otherwise, the blood system would be assessed for DNA. But I'll talk about methylated DNA and how we utilize these as markers. And so there's different types of cfDNA, and we've demonstrated all of these microsatellite mutation and CpG island hypomethylation and size in terms of DNA integrity. And they can be used for detection of subclinical disease or monitoring progression. So some of the early trials we did is biochemotherapy, where you do multiple chemotherapy pre and post surgery as part of an early program we did with this with Steve O'Day. Now this is starting to come more back into clinical practice, where you got multiple regimens now to where you combine surgery and neoadjuvant to more effectively control aggressive disease. And here's an example where we did pre-bleeds to predict overall outcome in stage 4 patients, and we show that if you're methylated for RAB beta 2 or RSF1A, you have a far worse prognosis versus if you don't. And on the right hand side, if you have both of them, you can see that methylated has a much more poor outcome in the first year versus those that don't. So here's a way you can predict disease outcome by monitoring the serum. Another marker that we came is estrogen receptor. It is found in melanoma in early stages, and then it becomes methylated. Similar to breast cancer, where estrogen receptor alpha is turned off by the methylation process. And this figure shows you how it progresses stage. And we were able to convert this to blood assays, and we can show that methylated is a far poorer prognosis than non-methylated. And you can see that. So this is a marker approach using the actual methylation DNA as predictors of outcome. And it's a new source of biomarkers if you find the right ones and how specific they are. So another type of marker that we looked at are called line 1s, long interspersed nucleic elements. And they're scattered all over the genome like alleles, repeat sequences. They're non-coding. However, they have a long history evolutionary-wise, and they recently have become more important, especially in solid tumors, that they are often inactivated, in other words, hypermethylated in normal cells, but in tumor cells they become hypomethylated, in other words, line 1 is active. And they are believed to play a significant role in genomic instability and also translocation. All our studies we do the laser micro dissection standard process, so you look at actually the tumor elements rather than the normal tissue to get real specificity. So here's we showed line 1 index of unmethylated, and you can see it progresses significantly from stage 1 all the way up to stage 4. You can see the activity is definitely turned on. And then here's another molecule that we looked at, was A1, a gene that was originally described in 6p21, and no one really has still found what it really functions, but we found that is methylated significantly changes during progression, as you can see on the right-hand side, that it becomes highly methylated during progression. And it may be an indicator. Still, we have not found what the function is, but this is an example. So what we did is, this is just before I get into that, this is an example of profiling, how we do it. What we do is we develop a way to look at overall methylation by screening the whole promoter region and sites next to the CPG island promoter region, which we call shores, and then even further, which are further out. And then we map out the methylation levels overall. And then what the colors are is comparing nevi, primary melanoma, lymph node metastasis, and MBM as brain mets and other stage 4. You can see the pattern. In the shaded areas is where the CPG island is the most influenced. You can see the brain mets are far more methylated than the nevi or the primary melanoma during progression. So you can see the difference, and this is how we overall profile the genes, individual genes. And then so what we did was combine stage 1 and stage 2 patients and looked at, these are primary tumors, and looked at whether how hypermethylation and hypomethylation of line 1, hypermethylation, hypomethylation A1, and or together, how they influence. As you can see, if you were unmethylated A1 and line 1 methylated, you have a far better prognosis versus those that are A1 is methylated and line 1 is unmethylated. And sample size is small, but you got to remember these are primary tumors that are only a few millimeters. And these are done off of paraffin section after microdissection. But what I want to emphasize here is two different pathways are going, the methylated non-coding versus the unmethylated methylation conversion during progression of the coding gene. And so this tells you that different events are occurring during progression and both in non-coding and coding, and they're quite important overall. And here's another example where we look at now stage 4, same way. As you can see, those with unmethylated A1 and methylated line 1 do far better versus those that are methylated A1 and line 1 unmethylated. So you can see it's quite relevant overall that these events continually occur during advanced stages of disease also. We have been focusing a great deal in my lab on epigenetic alterations in melanoma progression to the brain. As I indicated before, melanoma primary it's on the skin, but it can metastasize to the lung and predominantly to the brain eventually. In general, the longer the patient lives with, let's say, a lymph node metastasis, even though it's resected or other organ met, it eventually succumbs and brain metastasis eventually develop. And there is no real pathway that primary melanoma always goes to the lymph node. It can go, as we've shown, bypass right to the brain. That's where there is no lymph node metastasis or other organ. And so this is an overall profile where we did skin, primary melanoma, lymph node, and brain met, and overall whole genome analysis and methylation progression. You can see clearly distinct the epigenetic signature. We're using a 450 kb array assay. You can see significant changes from melanoma brain mets on the far right versus primary melanoma and more to the left side. This is a complex diagram, but it shows metastasis can go to the brain quite frequently. We show a circus plot, and this is basically on the top right-hand corner. It shows the difference between lymph node, which is the yellow, the inner part of the circle, versus the red on the outer. And this is the methylation level, where red is highly methylated. And you can see, and the outer circle is basically the individual chromosomes 1 to 22, and you can see how it significantly becomes in some of the chromosome regions individual highly methylated versus the lymph node. This is a good approach to demonstrate the methylation progression on individual chromosome sites. Underneath, you can focus on individual chromosomes. It's a diagram that shows how it changes from development from a melanocyte all the way to a melanoma, and you can see how they change for RASF1A on chromosome 3b21. And you can see how it becomes more highly methylated during progression. RASF1A is one of the really predominant genes that gets methylated in melanoma. It's involved in microtubule, cytoskeleton, migration events. It is found in other cancers too, like lung is well known to be active, and some in colon cancer. So one of the models that we've been doing with Isaac Witt's group in Tel Aviv, they have a melanoma, a human melanoma. They kept on transplanting it into nude mice, and then they developed brain mets, and they took those and retransplanted them and grew up until had clones that would always go to the brain versus those that always go to the skin or other organs. And we were able to use like these 3 different variants. And it was a very good model to demonstrate. And then we did methylation profiles, and you can see that we were able to separate out distincts among the variants, highly methylated versus less methylated, related to the individual clones, the ones that always metastasize to the brain versus those that only go to the skin. So this was a very good model to really assess it. These are the plots where you can see the distinct subsets, clusters of CPG island sites that they are clearly distinct. This is a more terrain graph, and it really shows you how individual clones have specific clusters of methylation quite significantly among the individual clones in comparison of one of the original parent clone, which is YBFRC, versus its other clones that metastasize in this distinct events. So this is the way how we can analyze epigenetic events occurring to specific organ metastases. Now I will switch away from melanoma and just give you a brief of what we do other analysis. And this is in colon cancer, and this just shows you just a overall view where the left side is the histology, showing A is the normal, B is the adenoma, benign, C is the cancer tissue. And on the right, what we developed, Aquama MINT assay. This is to methylated non-coding. Aquama is a very highly sensitive PCR directed to a specific MINT site where we have CPG island. That is, we know that's a regulator in methylation. You can see the adenoma is unmethylated, and in the benign, it becomes unmethylated and methylated. It progresses. So this is an example where we can really pinpoint very small areas from a paraffin section and really determine the status of the methylation of a particular site of the gene. Here's another example where we can do the same, where we look at these are tissue cores on the right, PMAs, but where we stain them for in colon cancer, which is MLH1, which is a stability factor regulator gene, and PMS2. As you can see, we did the methylation either by CEQ, capillary array electrophoresis, and you can see unmethylated where it's active, and you can see the staining on the top two right-hand side, but then you get methylated, it decreased significantly or no staining at all for HMLH and PMSC. So here's another way of showing how what we do is correlate IHC with the MF, which is actually quite important to really demonstrate. You have to have both to really show that the unmethylated or methylated events actually correlate to the expression of the gene. Here's another example where we looked at COX-2, which is a very well-known inflammatory gene, and it's found in GI cancers. Particularly, we looked at primary gastric cancer. Here's another example. On the right, we do capillary array electrophoresis. You can see it's unmethylated. On the left-hand side, you can see it's well expressed, staining. And then it just shows in a large, this is from the Dutch study by Cornelius van der Wel, where he shows the long famous trial, the D1, D2 resection, regards to lymph nodes. It has a long follow-up of over close to 15 years now. And we had these specimens, and we assessed to suggest whether methylated COX-2 or non-methylated has any event. And you can see the methylated, which is gene turning off of COX-2, have much better survival than the non-methylated. And traditionally, people have done IHC, but IHC is often difficult to interpret. And here we show we can convert it to a DNA methylated. And you can see it is actually a real mechanism. And overall long-term survival, you can see the difference in this trial. Another example that we've been doing in rectal cancers, where we looked at retinoic acid receptor methylation and other transcription factors, MINT, MINT31. We find, and we looked at rectal cancer, particularly from the TME transmesial rectal trial, which is another large Dutch rectal study, which is basically a pillar of many of the current trials in terms of treatment of rectal cancer. And we looked at that and demonstrated it was significant. Here's another study that we did on breast cancer, primary breast cancer, where we did methylation status, and we were comparing ER negative and ER positive. And this is a study recently published where we looked at this large cohort of patients that were on hormone therapy and no other adjuvant therapy, and we looked at primary tumor, and we divided into no methylation versus intermediate methylation versus high methylation. As you can see, the high methylation, the red in the top right hand corner, the worst overall outcome, same as on the bottom. You can see the disease concurrent, and on the right hand side, overall survival. And you can see there's a 15-year follow-up on these studies, so these are well documented. Here's an example. Then we started dividing the ER negative versus ER positive in these tumors, and there are many curves here. It's a little complex, but basically the pattern still falls on the ER negative, the ER positive. They're different, but if you look at the individual groups, on the bottom left-hand corner in particular, you see ER negative, which is blue, which is normal. You get a very good survival rate. ER negative, intermediate, you get red line, which is poor survival, and then the orange is very poor, which is highly methylated. You can see a significantly different in these Kaplan-Meier curves. There's a significant difference. ER positive does the same thing. So this is a very in detail analysis of ERs, positive and negative patients, but looking at specific these methylated markers that we defined to predict outcome long term. So in summary, overall, I've given you a synopsis of tumor epigenetics. They play a significant role in tumor carcinogenesis and as well as tumor progression. They regulate gene transcription, that's mRNA levels, and can turn genes on and off, just as powerful as deletions and mutations. Probably more common than other events, especially in cancers, since they affect many genes. They can be used as diagnostic and prognostic markers, as I showed you, both in tissue and as blood, as cell-free DNA. They can be reversed by drugs I didn't show you, 5-azacitidine. Some of the newer drugs are the methylation of promoter regions, but actually now some of the HDAC inhibitors, which affect histones, can also reverse. So it's very promising in the future. So my studies are supported by program project, R01 grants, and private foundations, such as the Addison Medical Foundation and the Yale Foundation. Thank you very much. If there's any questions, please post them. All right. Thank you very much for listening. Any questions you can post, and hopefully I can address them in the future.